Okay, welcome everyone. This is the Physical Samples and Collections interest group. And our session today at this plenary is on sample identifier and metadata practices tools to enable interdisciplinary sample data discovery, integration and reuse across multiple systems. Unfortunately, our senior chair, Kirsten, could not be with us. Esther couldn't make it either, but Jens and I are here and we're going to be ably supported by Joan Demerose, Sarah Ramdeen and Steve Richards. Next, please. So welcome. Our session is being recorded. Please mute yourself when you're not speaking. And please fill in the collaborative notes document. You can see we've Stephanie's been posting it. And also we've just added a column to try and understand what sample types you're interested in, because every plenary, the diversity of sample types just keeps growing. So if you've got any questions or comment, please use the chat or the session document. Next slide, please. And so what is the rationale of this interest group? It's about reproducible sample data, reusable samples and data, and recognition of sample collection and curation as a scholarly contribution. So that is ensuring people get citation and credit. And we cover a lot of topics ranging from practices, standards and policies, including persistent unique IDs, metadata and terminology, registries, catalogues, harvesters, sample sharing, preservation policies and practices, and most important, physical infrastructure for sample preservation and access. Next. We've been going for quite a while, starting in 2017 at Montreal, where we started our interest group, and we've been running sessions at almost every plenary ever since. And again, if you click on any of those links, and by the way, there is a link to this slide deck in the notes page, so you yourself can pick up on all the links. And now we're at P18 in November 2021. So it's 4 years we've been going. Next. And so for this session, we want to provide a forum for existing and emerging initiatives related to physical samples in the research data ecosystem to update the community on their goals and activity and work with each other and other RDA interest groups for a broader, to make sure we're getting samples fitting within the broader research data ecosystem. And as I said, we want to get a shared vision on the strategy for advancing discovery, accessibility, interoperability, and reusable of materials samples across domains for the benefits of science and society. Next slide, please. And so this is the agenda. I'm just doing the intro. Esther Plomp, one of our chairs, has got the 23 things, physical samples to a mature stage, and that's now out for comment. So we really need people to pay attention to it. And then we've got a series of 5-minute lightning presentations on some major systems. And then we'll go into a deep dive, which will be divided into 3 concurrent breakout sessions with a circle back and synthesis. Next. So there's 2 documents. The first is the attendees list. And the second one, I'll just put, this is something new we've just started for this session. And it's a crowdsourced list where we invite you, if you've got a project of any sort, please put it in that list. And we will be running this now regularly for each of our sessions. And we don't care how big or how small your project is. If you're dealing with something where you're getting samples, getting them identified and curating and doing anything, please put it in that list. and we can start to get an idea and maybe get better collaboration going between the projects. Next slide, please. And so now I'll hand over to Sarah. Can you run Esther's video on where we're at with our major output, which is the 23 things for physical samples. And as I said, it's out for community review, so please contribute to it.
Hi everyone, my name is Esther Plomp and I am one of the co-chairs of this interest group. And as one of the co-chairs, I would like to give you a brief update on what the group has been doing the past year, or since the last plenary that we've had. I am unfortunately not here live in person, but I'd be happy to answer any questions you have. So do feel free to reach out to me if you have anything that you would like to discuss. So one of the things that our group has been up to is provide 23 things for physical samples in a very useful, user-friendly flyer, which basically gives an overview of all the relevant resources that are of interest whenever you want to do anything with physical samples. So if you would like to have a look at what the community has been setting up for this, you can now see the output because it is under community review. So you can go to the link in the slides, and if you have any comments, input, we would still really like to hear from you. So please have a look and provide your input through the link in the document. We've also been hosting a webinar series together with ESIP, and we've done 3 webinars. And the first one was on electronic lab notebooks and was primarily focused on RSpace. And the recording is available on the RDA website. So if you follow the link in the slides, you can go to the more information about this webinar and rewatch the whole thing. Our second webinar was on RRIDs, which are a way to track regions through the literature. And our third one was on a very similar topic as our current session for the plenary on supporting interdisciplinary sample discovery, integration, and reuse. So we hope you've been joining these webinars, but if not, the recording is still available and you can still have a look. And if you're watching the recording from the plenary session instead of joining it yourself, I would still encourage you to join our interest group so you can stay up to date with the latest developments regarding physical samples. And we would love to have your input, so please do join. Thank you.
Okay, so can we move on to the next slide, please? So now we're going to have a series of 5-minute lightning presentations on various major systems developing infrastructure for samples. Okay, so I think, Sarah, you're up first. Yes.
Yeah. So hi, everyone. My name is Sarah Ramdeen, and I'm co-chair of the ESIP's Physical Samples Cluster. We wanted to share a little bit of what we've been doing, especially because of the close connections we have with shared communities and interest, as well as shared membership. Sorry, it's hard to advance the slides and talk at the same time. So the ESIP is the Earth Science Information Partners, and the cluster goals for our particular cluster are to review existing tools and services from the community. One thought we had was, instead of constantly reinventing wheels, is to give people the opportunity to see what else exists out there, and then what can we redesign or repurpose for new paths forward. The other goal is to provide a space to discuss methods for streamlining workflows. We have a lot of repositories that are part of the ESIP community that work with individuals either loaning out samples or acquiring samples from researchers, and the discussion of workflows within that network, as well as connections to publishers and the research community beyond, is of important interest to us. And And the final goal is to connect with external partners and organizations similar to the goals of this interest group that are trying to engage with physical samples, so that again, we're not duplicating efforts, but we can share burdens and also learn from each other. Our current activities include first this joint webinar series that Esther just spoke about. I've got the links to the YouTube videos here as a shortcut if you just want to get to them that way. We're currently pausing the series just because of the holidays and AGU coming up, and then ESIP in January, and we'll start again in February. If you have any topics that you'd like to see or a presentation you'd like to give as part of the series, I would encourage you to reach out to one of the chairs of either the RDA cluster, interest group, or the ESIP cluster. Our other current activity is that we're working on guidelines for publishing samples and sample-related data, specifically with the idea that it would be guidelines hosted by journals recommended to authors. It came from a variety of needs. First, we saw this as a need in the community where people were coming to the cluster with these questions of what do I do and how do I do it, and what are maybe some very basic recommendations. And then we also had a presentation from Shelley Stalls at one of our sessions, who also said that AGU was looking for recommendations that they could host themselves. So we started as a cluster where we just reviewed possible templates and structure so that we could host these guidelines in, at first trying to keep it to something very brief, 2 pages, which was kind of impossible. And then we also reviewed the scope of the content of what these recommendations might include. And from there, we started brainstorming a document, and we were given as an example template from AGU this data and software sharing guidance for authors submitting to AGU journals. And it's a recent publication, and I meant to include the link on the slide. I'm sorry, I forgot. But we thought that would be a good first base because it has a lot of good similar content that we could either reuse the structure of, or also some of the tips and guidelines, but also then understanding that sometimes these things don't work for samples, and that's why we have our own communities and clusters, and so that we might rewrite or change those things to fit our needs. So the cluster meets monthly, and where we've been working on this, and then we'll also be hosting a session at the ESIP January meeting to focus on continuing efforts and hopefully getting a draft of this document out. Once it's complete, we'll send it out to the community for review, and hopefully we'll target this community as well, the RDA community, to get feedback. And then once that's done, what our vision is that ESIP is very, again, earth science focused, but there are many other types of samples, and that there's the hope that the guidelines might be picked up and adopted and modified by other communities to fit their particular domain needs. So there's a number of ways how you can engage with the ESIP Physical Samples cluster. It's open to anyone. You can sign up for our listserv, which is the first link on this slide. And then, as I mentioned, we have monthly telecons, which again, open to anyone to join. They're the 4th Tuesday of the month at 5 p.m. Eastern. The next one is November 23rd. And you can get information about how to join the Zoom calls from our wiki linked here on the slide. And then again, we'll keep in touch with you guys as we make progress on the guidelines. And I think I'm the next talk too, actually. So yeah. Leslie, did you want to say anything before I moved into the next one?
No, we are now moving on to 2 projects which Sarah will present on the eye samples and sampling nature. Go for it, Sarah.
All right, so today I'm presenting on behalf of Kirsten Leonard, who, as Leslie mentioned earlier, is unable to attend, and I'll be talking about the eye samples and the sampling nature projects. So these projects aim to support the transdisciplinary use of natural history material samples and to realize the idea of sample access, sample discovery, and sample interoperability. This talk in particular is a set of highlights from a longer talk, which was part of this interest group's webinar series. So you can find the full talk by following the link at the top of this slide. So there's an increasing need in the sciences to study samples across disciplines. There are a few examples listed here on the slides, and I won't read into them, but they include soil, ocean water, pottery, or rock samples. And these things can be collected for a specific purpose, but then can be used by many other researchers, even in other domains, in order to do other science. And the project aims to support these efforts. Eye samples and sampling nature are separate but closely connected projects. Both projects are funded by the National Science Foundation here in the United States. iSamples is the technical side and aims to build the cyber infrastructure for managing samples across domains. And Sampling Nature is the community aspect of building a research coordination network that spans a wide range of domains and provides scientific vision of working across those domains. It will develop and promote best practices standards as well as to help overcome the social barriers within this work. So the approach, there's a lot to it, so I'm just going to give a summary here, but the approach of both projects is to create something that is flexible and scalable to work across domains, both on the technical as well as the implementation side. So this is to ensure that it can be broadly adopted by diverse stakeholders. And the project team recognizes that there are system practices that are already established and deeply embedded in the community that we do not replace and that we do not want to replace. These projects specifically want to integrate the established discipline-specific infrastructures and to expand the capabilities, but also to enhance the consistency of these infrastructure projects and thus their impacts. The project aims to work with and promote high-level metadata standards for natural history standards that work across biosciences, geosciences, and archaeology, while also supporting the community-developed, more detailed metadata standards for these specific domains, which Steve Richards will talk a little bit about coming up next. The projects will seek guidance from the International Sample-Based Research Community, Natural History Value Chain, and Sustainable Science.
Sarah?
Yes.
Sarah, your audio is getting very bad and there's a lot of background noise.
Outside at a workshop. I don't really know how to solve this. I can try moving inside, but that will take a moment.
No, it's okay. There was just a bit missing when you were talking.
I think I was moving on to the next slide. All right. So the overview of these two projects shared here is very high level, and we encourage you that if you have any questions or want more information to reach out to us. But from the cyber infrastructure side, it consists of two components: the iSamples in a Box, which is a standalone software package that interested institutions or organizations can deploy. And it creates an identifier and stores associated metadata. And then it synchronizes those with iSamples Central. iSamples Central is an aggregator of metadata that provides discover and resolution services with an API as well as online search interfaces. iSamples Central provides a gateway between iSamples in a Box and the different identifier authorities. So no one who uses iSamples in a Box has to dedicate themselves to one identifier, but can choose which identifier they want to use. The iSamples project will have 4 use cases for the iSamples in a Box, which include CSAR, Open Context, Geome, and the Smithsonian. Finally, the Sampling Nature RCN aims to bring together international and interdisciplinary groups engaging across multiple communities and agencies. The activities are a set of working groups focused on standards, policies, uncurated sample collections, and training materials. We will also engage the community through workshops that focus on the Sustainable Development Goals of the UN and topics brought forward by the working groups. The deliverables for this project include extensible community-driven metadata standards and recommendations for its short-term and long-term governance, as well as policy recommendations, training materials, and an increased awareness in the community about the value of material samples. So if you have any questions about either projects, I've included the websites on the slides, and you can feel free to reach out to any of the co-PIs. And now, Steve.
Okay, so now we'll pass over to Steve, and he's going to present on some work that's come out on metadata mapping with iSamples across multiple systems. Right, go for it, Steve.
Thanks, Leslie. So I'm working on the iSamples project, and we've been focusing on the metadata project, the metadata scheme that we'll be using. So the objective here is to develop a core or kernel sample description scheme that's applicable for material samples from any of the partner systems that we're working with. Sarah just mentioned that. So the approach we've been using is to look at the existing sample metadata from our partner projects, and that includes open context with various kinds of archaeology related samples, including things like pot shards, bones of various sorts, other kinds of artifacts, genomic samples from Genome. CESAR, the Earth Sample Registration, Earth Science Registration, has water, rock, gas, experimental results, physical samples, and from the Smithsonian, which has a very wide variety of collections, mostly biological samples. And in addition, we reviewed existing models that we could find out there for sample metadata, including the IGSN metadata, GeoSciML, CESAR, the digital specimen activity that's going on in connection with TADWIG and GBIF, ODM2, and the W3C SSN model, and also ESS Dive. So by looking at all these things and basically looking for the commonalities between all these, the metadata schemes that are there and the content that's in the metadata that we're actually working with, we came up with a draft scheme that we're using, and we've been testing. We've harvested now metadata from all the partner systems, identified a few problems, but I was just going to briefly flash on the metadata framework that we're using. So first of all, in terms of identifiers, we're working with, there are two things that get identified. One is the actual material sample itself, the physical thing in the world, and that has an identifier. And on the web, of course, you can't transmit a physical sample. So when you dereference that identifier on the web, what you get is a sample metadata record, which has its own identifier. That's over here on the other side of this diagram. The thing in the information system is the sample description, also known being called the digital specimen, which is the representation of the physical thing in the information world. And it has a separate identifier because it's managed separately. It's a different resource. So there are these two critical kinds of identifiers that we have. Then in terms of the metadata content, there's some basic discovery properties, a label, so the human intelligible label, the sample identifier typically assigned by the original collector in many cases, which generally is not globally unique, and also hopefully some kind of description of what was collected and some information about why. And these things are very useful for indexing in discovery systems. Then we developed 3 controlled vocabularies for high-level categorization of samples across domain, and these include a specimen type. So what kind of thing was collected? What is the actual object that you would see on the shelf in your sample collection? The material type, what is it composed of? And then the sampled feature. So what is the sample intended to represent? What was the context from which it was collected? And this is sort of goes back to the idea that sample is a thing as a sample because it's a role. It's intended to represent something in the world. And then in addition, there's keywords, and this is sort of a free for all tagging scheme that the sample collectors or registrants can use to help with discovery. Then on top of that, then there's all the other stuff, which is mostly related, or in large part related to the reuse part of it for evaluating, is this sample useful for the purpose that somebody else might want to use. So what the most important thing is probably the sampling event. So this includes information about where was the sample collected, how was it collected, who collected it, when did they collect it, curation information, where is the sample now, and what are the conditions for accessing the sample or using it to do other research. Sorry. And has the sample been modified since it's a session into a collection? There are all kinds of relationships between samples and other samples in terms of parent and child relationships, derivation, where samples are derived from other samples, but also relationships between samples and publications that are both about the sample or publications that are based on data collected from the sample. Then there are data sets that might be based on samples. Samples can be members of collections for various purposes, for a collection related to a research project or any kind of contingent collection of samples for some purpose that you might want to relate a sample to. There's some other properties are depending on the different domains we might be interested in. and these can be assigned just using key value pairs. Of course, you need a vocabulary then for the keys for the properties that you're interested in assigning to. And there's also a variety of other kinds of categorization schemes that are important in different domains for describing a sample. So things like in geology, you'll be interested in rock type. In biology, what's the taxa? In archaeology, what's the cultural context for a sample? So these all have associated vocabularies as well that might be interesting. So those would be part of a high-level scheme. Got to press the right button here. So I just put some links in here. I won't review these. One thing I'd like to point out, though, is we set up these card sorting exercises to test the vocabularies. And if anybody's interested in checking these out, what it is a collection of sample descriptions, and then the card sorting is trying to read the description and assign it to one of these high-level categories that I mentioned that we have the controlled vocabularies for. And we've been using this to sort of test how workable these vocabularies are. All the work on this is in GitHub, and there's links there. There's a JSON schema for the metadata, and also information about the vocabularies, both in a decision tree for understanding the vocabulary, but also SCOS files that contain the vocabularies. So I just like to put in a plug for the breakout session. We'll be addressing some of these questions. If people are interested in looking at what's in these material specimen type or sampled feature type vocabularies, we could look at that. Thinking about what elements need to be in the kernel. Are there things missing? And also, if people are interested, we could look at the implementation in JSON LD and have discussion about that. So that's the upcoming breakout session. Thanks.
Okay, thanks very much, Steve. I'll now pass over to Jens to give us an update on what's happening between IGSN and DataCite. Jens, can you take over, please?
Yes, thank you. So I want to give you a brief update on what is happening between IGSN and DataCite. About 3 years ago, the IGSN implementation organization started a project to re-evaluate its business model and its technical architecture to make it more sustainable and to operate it into the future. And this is why we dubbed this project the IGSN 2040 project. And one of the primary findings of this project was that the IGSN implementation organization should focus on scaling its activities on community engagement and development of sample identifier practice standards, and encourage the adoption of these systems through third-party implementations and platforms, but outsource the running of the technical infrastructure to a trusted partner. So we held a number of partnership conversations, and then in the end, DataCite and IGSN came to a very good arrangement that it's now formulated in the IGSN DataCite Memorandum of Agreement, which describes this partnership in which DataCite will be running the ID registration service, and but the way it's set up will allow IGSN to maintain its identity and brand and free resources for working with the samples community. There's some more documentation that the announcement will point you to that outlines what this community engagement might look like. Next slide, please. So the key details about this partnership is that IGSN will transition to the DataCite infrastructure and services, and using the DataCite APIs. What is also quite exciting, I think, is that new and existing DataCite members will be able to adopt and start registering IGSNs within their existing DataCite workflows and using the IGSN trademark. So the benefit of this is that you don't need to run two separate infrastructures to mint DOIs and IGSNs. You can do it all through one infrastructure and one account. The existing IGSN IDs will all become DataCite DOIs, but the handles server that we are running will continue to operate, so that existing DOIs will be able to resolve into the future, at least for a long time. We will decide how long that will be, but it will be many, many years. In this agreement, DataCite will be responsible for hosting the technical infrastructure and registration service and this legacy IGSN handle system. And the IGSN implementation organization will then continue to maintain and grow the international samples community of communities that uses persistent identifiers for samples, and coordinate a cohesive use around IGSNs. And then lastly, and not least, the IGSN implementation organization's community of communities will provide a space to promote the adoption of fair and care principles for physical samples. Next slide, please. So this is a cartoon overview of the transition timeline of how we will move from the present day IGSN implementation to this implementing IGSNs in this partnership with DataCite. We are at square one at the moment, defining the timeline, and moving to square two or point two, where we are engaging with our existing IGSN allocating agents to discuss this transition to DataCite. In the next year, we will be working on transitioning the IGSN resolution, working out more details of the technical plan for this migration, and start moving agents over to the DataCite infrastructure, and probably also start aliasing the IGSN handles into DataCite DOIs, so that both the legacy system continues running and is also resolvable in the new system. And then in the year following, we will work on enhancing the discoverability of samples and support and scaling the operations and working with the community to grow the use of IGSN identifiers for physical samples. Thank you.
Okay, before we go into our deep dive, I'm just wondering, we're actually on time. Does anyone want to ask a question of any of the speakers, that is Jens, Sarah, or Steve? Okay, so someone's put your hand up. Who is it?
Steve.
Steve. Hi, Jens. Question, what about the metadata scheme? I mean, the DataCite, the DOI identifier scheme has a fixed DataCite metadata scheme that doesn't really, I mean, there's been some discussion about this, doesn't really have some of the things that we'd like to have for metadata about samples. Will there be some way to distinguish DOIs that are about physical samples from DOIs that are about publications?
Yes, that's an excellent question. It works on different levels. So the immediately distinguishing feature between different DOIs will be the element that describes the resource type. So DataCite at the moment has resource type other for physical samples. It's miscellaneous in the present system, but it will be physical sample or physical specimen in the future. So this is a very easy way to distinguish what kind of resource you're looking at. Also because they're taking other communities on board, like for instance, persistent identifiers for instruments. So you will be able to distinguish a gray literature publication from a data set, from a sample, from an instrument, and other objects that they might be identifying in the future. In the first step, we will map the existing IGSN description metadata onto the DataCite mandatory and recommended elements, just for initial interoperability. Then DataCite is reviewing their metadata schema for releasing a new version, and we have been invited to contribute to that. And then there are some ideas that came out of the IGSN 2040 project to approach metadata description and harvesting and discoverability in a very different way that will be way beyond the scope of this session to explain that. That is further into the future to think about how we can scale these systems to beyond the millions of objects that we have now to billions of objects in very diverse settings, and still have the system in a way that it can serve the specific requirements of communities and also interdisciplinary communities. So schema.org is a keyword that I can drop on that. So you can see that 3 different horizons: the immediate interoperability, second step, expanding the schema, third step, going to something completely different in the future.
Right. Huh. Schema.org will need some updating if it's going to work.
Oh yeah.
That's a harder row to hoe than DataCite, I think.
Okay, just quickly, Robert Hanisch has said DataCite has 1 million samples. I think that's correct. Jens could say otherwise.
No, that is correct. It's mainly from the Food and Agricultural Organization.
Yeah, IGSN has 10 million.
Mm-hmm.
Yeah, as we said, DataCite is starting to have research objects in it, like instruments, the RAID, the project IDs going in, and we're going in now as physical samples. So there's a few changes that are happening. And Taco has asked, Jens, if one is going to start with sample IDs, what would you advise to use, IGSN, DataCite, or iSamples?
I think that IGSN and DataCite will be the same from now on. So that question is settled. It's DataCite, and then iSamples is a particular way of bringing the community together and equipping them with resources. That's what I understood from the presentations. So what I understood is that iSamples is somewhat identifier type agnostic. So of course I would advocate for using DataCite DOIs, but if your setting requires something else, then iSamples would accommodate that.
Okay, thanks, Jens. We'd better get going because we're 4 minutes behind schedule. I'm now going to hand over to Joan, who's going to lead our deep dive. And from the deep dive, we will then have 3 breakouts. So Joan, can you take over, please?
Yes, thank you, Leslie. So I work in community engagement and standards development for the ESS Dive data repository. And this is the primary repository for the United States Department of Energy's Environmental System Science program, which is a interdisciplinary research program. And for this deep dive, I'm going to present work that we've started in collaborating on use cases across 5 different data systems that support the Department of Energy's biological and environmental research. Next. So this environmental system science program includes really diverse data that spans a variety of different kinds of observational, experimental, and modeling activities in earth and ecological sciences. And at ESS Dive, we're working to support scientists in their data publication and also to develop standards and tools to support their research, which is really highly interdisciplinary and collaborative, uniquely so. And there's often really large multidisciplinary teams coming together with a variety of different data types that we need to link and exchange information about. And so work often involves a variety of different biological and environmental samples to explore questions in interdisciplinary areas like watershed sciences, terrestrial ecology, and coastal systems. And we've also found that more work is really needed to effectively link related samples and publish data sets and methods, projects and journal publications, all these things that contribute to research that may exist across a number of different data systems. Because often these research entities are currently pretty disconnected. Next. So early in our community activities and discussions with scientific projects, we found that they were facing a particular challenge in efficiently tracking samples as they're sent to different collaborators and labs and facilities for a variety of different physical, chemical, and biological analyses. And through our research and pilot work, we found that integrating sample data effectively for these kinds of projects as data is added over time and transferred across numerous different data files and then published in different online data systems, it really requires using globally unique and persistent identifiers, which nobody was really doing. Next. So we decided to work with a number of different projects on using IGSNs to link this related samples and data. And we did this through by working with our scientific users on standardizing their metadata and assigning IGSNs through the System for Earth Sample Registration, or CSAR, which minted the IGSNs, of course, and maintains the persistent web links, which is really valuable for us because we didn't have that capability, and also provides a number of useful online sample management tools. Next. So in the case of DOE's biological and environmental research, scientists are often working with or across potentially 7 different data systems that we identified through our use cases that are shown here. And these are all dealing with different aspects of sample data analysis or publication. And so the persistent identifier, the IGSN, really was the way to provide a way to link and exchange information across the facilities and data systems. And if we did this on a broader scale, it would improve our ability for people to find, compile, and reuse interdisciplinary sample data. But we also found that there's a lot of work to do in coordinating across existing systems. For one, to make sure, like as we're going through these use cases, that we understand the science need and the most useful entities to link. The metadata that we need to do this effectively, like relationship terms, and work together on common protocols to exchange information across all of the different systems. Next. So we have started working through project-specific use cases to add URL links with descriptions on associated relevant landing pages. And the purpose of this is to really test out the specifics on how we can usefully associate things like the sample landing page and the ESS Dive data sets in a meaningful way for scientists to associated data in resources such as the Genome's online database and many others. And so our goal in this work is to eventually figure out how we want to add related links automatically using the DOIs and IGSNs and the associated metadata to make publishing this interdisciplinary data easier and to make the published data more complete and usable. Next. So a critical step in this work is to coordinate across the relevant data systems and standards groups that are involved in publishing the metadata and data on both the environmental and the associated biological samples that are often extracted from, for example, like a soil or water sample that was originally collected in the field. This coordination includes a number of different labs and facilities that analyze and publish the data, the biological omics data, and then also environmental data repositories such as ESS-DIVE, the Genetic Standards Consortium, which developed the minimum information about any sequence template that is required of people who are submitting genomics data, and then other groups such as ice samples. And so one critical issue that we're currently working on as a starting point is linking a source material originally collected in the field to associated biological extractions. And within the minimum information about any sequence, that is the source material ID. And we are starting to work with the Genetic Standards Consortium and groups such as NCBI on a standard guidance for providing a source material ID that is actually a persistent identifier, because they don't have that capability currently. Next. So our goal is to work on a standard approach across systems to link related research entities, including things like samples and then related data that's held in other repositories, and then even methods and journal publications using related identifiers. And so in the breakout, we'll discuss proposed metadata needed for this, which addresses the sample metadata and data access and linking across data systems. So this is really a key first step that most data systems are not doing currently, and more work is needed to agree on a standard approach. Next. So this is just the final slide. So coordination really across existing groups that already have established practices. It's not really, hasn't been easy, and it takes both advocates and time, but this is a really critical step in that we need to work together to basically improve user experience for scientists who are using persistent identifiers for samples, to make sample tracking easier, to make better tools so that we are able to get higher quality metadata and data, and just generally more easily be able to compile and link related samples and data across systems. Next.
Okay. Yeah, all right. So yeah, just as I said, we'll now go into 3 breakout sessions, and each one of the leaders will just do a quick recap on which the breakouts are that we're running, and we've asked them to give an elevator pitch in a building that's only 1 story. So it's an elevator pitch for you to listen to as you go from the ground floor to the 1st floor. Okay, next. Steve, these are what you've said before. Is there anything you want to add?
I don't think so. So we'll be looking at the metadata.
All right. Next slide. Joan, your elevator pitch.
Yeah, I just mentioned, we'll discuss in more detail the coordination efforts, identifiers, and metadata needed across data systems to link and exchange information on related samples and interdisciplinary data.
Okay, and next slide is me, and I'm just trying to, we've just put this in as a suggestion, in that we're doing all these things to get samples identified and linking, but there's a fatal flaw in that people do not value physical samples in the current environment. Many collections, as we know, are under threat and being thrown out. And I just went down a trip of memory lane where in my university department, way back in the last century, we had a full-time museum curator And I don't think many schools even have that in this day and age, let alone have a place where there is a collection. And so I'm just going to have a bit of a debate. What do we need to get samples valued so that we'd then take off and do things like what Steve and Joan are talking about? Who needs to get the credit? Where do we need to cite samples? And where can we best focus our efforts to make people aware that physical samples in research are becoming a threatened species? So next. Next slide. Okay, right. So what we're going to do now is if you click everyone on the breakout rooms down the bottom, on the bottom bar, and you can click on the assign to and pick which group you'll want. So group 1 is interdisciplinary discovery with Steve. 2 is metadata access and linking led by Joan, and 3 is reuse and credit. Now each group has its own breakout sheet, which the leaders will help you with. And what I'd ask is, can each group leader come back for the circle back session with 3 to 5 key takeaways? Thank you. Stephanie, are you there?
I'm here. Yep. I opened the breakout rooms and
Can you put me in reuse and credit, please? Because I can't see my name.
I can try. Hang on.
This is what happened last time.
Oh, it's good. Reuse and credit?
Yes, please.
I have assigned you. Anyone else who needs to be assigned somewhere? Welcome back, everyone. Leslie, are you good to take over?
Yeah, I think it was.
Who's on first?
I don't know. Leslie, you're muted if you're with us.
Okay, I'm unmuted. Right, Steve's on first. What are your three key points?
3 point. I don't know if we have 3
Oh,
points.
hi, Kirsten. Hello, here's our other
Hey,
leader
we're
who's
actually
been missing.
together here. First in-person meeting.
So we have discussed, we had a discussion about the utility of a high-level controlled vocabulary for categorizing samples in a cross-domain sample registration system like iSamples. And it's the, you know, the sort of question of is it better to let people use, you know, strictly use the terminology that they're accustomed to using in their community, that they're comfortable with, and it doesn't require a lot of effort on their part to figure out. On the other hand, by having these, what we were thinking in the iSamples was if we have some of these high-level controlled vocabularies, people will have to understand what they mean to use them, yes, and there's definitely some education on that side from the data providers. But the idea is that for the data consumers, it gives them a way to, if you're searching the iSamples catalog, let's say, and it might have, you know, literally millions of samples in it, it gives you a way to prune that universe down to narrow your search from an early stage. And I think, you know, there are arguments both ways, and it was a good discussion. And I guess my takeaway is we really need to get the user interface up and running better to actually find out, do people use it, and do other people find it useful for working with. And I think that the jury's out, and it was a good discussion. Anybody want to add anything?
Where did Jens disappear?
He's there.
Oh, there. Oh.
Okay, Joan, would you like to report back on your key takeaways from your breakout, please?
Sure. So one of the things that we started with was just other existing use cases or approaches to link related samples and other research entities like data and data sets and publications. And one thing just that came up is that in the biomedical community, for example, there were Maria, who was in our group, pointed out like a tissue repository that was very important, but the data wasn't linked at all or not associated with metadata. And so there were examples where groups could be doing this but aren't. And then Fran was also in our group and brought up the USGS, some efforts there to improve documentation of laboratory procedures involving samples. And there's a particularly good example of involving the International Coalition of Curators on seafloor cores, where they are starting to link related data more. But in general, for coordination efforts, we really need to work on developing infrastructure that makes it easy for people to assign identifiers and assign identifiers and to have databases available with information on the relationships. And I think there are a lot of great projects doing this already, such as iSamples, getting that kind of thing going. We also talked about like what metadata or standard protocols would be needed to link and exchange information about related samples and data. And so just we reviewed some of the current metadata that is outlined as part of DataCite of related identifiers, and providing information on identifier types and URLs, and like related resource types and relationships, and information on exactly what you're linking that you can provide on a UI so people know what links they may be interested in. Some additional information that we might consider are things like the provenance ontology and linking samples by locations or collection sample sets or collections. And then in general, for actions that we can take to coordinate better across groups, we kind of agree on that starting with use cases is really important, specific use cases, and go through exactly what organizations and data systems are involved in how we link related information, and then working with communities such as RDA and ESIP on outlining these specific needs.
Okay, well, thank you very much, Joan. I'll just go on to my group, and we were talking about, you know, giving credit for both the funders and the person that collected the data and what are the roles that are associated with it. So we looked at some work that's been going on in the ESIP group on research object citation, and that had looked closely at the credit taxonomy, and that group had already concluded that the credit taxonomy did not work. We also briefly looked at a crosswalk that Ted Haberman has done on roles for citing roles in data, which goes across 19115, DCAT, and I can't remember what the other ones are, but it's got about 4 or 5. But accepting that the credit taxonomy does not work for samples, what do we do? And the suggestion is that maybe as a subgroup of this interest group, we could go to the credit taxonomy and actually say, is it possible for us to modify or add roles to it that make it more suitable for citing the people who have played a role in getting the collecting the sample and preserving it. But the other thing we also considered we had to take into account was the funders and the grants. And so if we can actually say this funder and this grant supported the collection of this sample, then you can see how Joan's work takes over, because then you're linking all the subsequent work that's done on that sample, including it being cited in a research paper. And so you can see from that you're starting to identify and build up a value role or value proposition for a particular sample that theoretically you could take back to the institution or the funders and say, look, this is the impact of scientific research on you funding the collection and curation and storage of this particular sample. Shelley, there was a bit going on in the chat. I didn't quite follow it all, but did you say you've already contacted credit, or where are we at with that, please?
I think Sarah said she did, but I am on the steering committee for the NISO Plus next meeting in February. And so we're, you know, should Sarah not get a satisfactory answer, I can support additional query.
Okay, so that's again just throwing that out as to whether this group pursues it. It is on the cards for the ESIP data citation cluster, object citation cluster, but the catch is they're currently much more focused on citation of data, and again doesn't cover the broader range of samples. It's earth and environmental sciences, whereas you've only just got to look at the fields that people filled in this attendance sheet to realize how broad the sample types are that are covered in this group. Okay, so does anyone from any of the groups want to make any other comments, please? Hands up. Okay, so for the next steps, I'll remind you that we need to review, golly gosh, where's the chat gone? Okay, so I'll put the 23 things. So that's open to the whole of the RDA community, 23 things for physical samples. If you want to change or review it, please look at that. Also, please get in touch with the team if we will resume those RDA ESIP webinars next year, which is not all that far away. If you have a suggested webinar topic or would like to give one yourself, please contact us. And then, oh yes, Stephanie's put in the chat that the 23 things is open for comment until the 2nd of December. And above all, if you're new to us, here's the link to our web page. And here's the link to the group's webpage, and from that you can get the link to join this cluster if you would like. Okay. Now, last call for any comments from anyone. I'd like to ask Joan or Steve or Jens, do you have any comments as speakers? And Sarah?
Not me. Interesting session. Thanks.
Joan, do you want to do any follow-up?
Just if anyone is interested in discussing more about a standard approach for linking samples and related data, you can contact me, and I'm putting my information into the agenda.
Thanks, Joan. Sarah?
I just want to thank everyone for the, sorry, we're getting feedback. Anyways, for the good contributions to the session, but very much looking forward to any of you joining us in the ESIP physical samples cluster and helping us with this journal document guidelines.
Okay, and Kirsten, are you still here or has she disappeared? Kirsten, do you want to say anything?
As I only attended a shorter part of this meeting, I don't feel in the position to make big comments. It seemed to be a really good conversation, and I can only say I advertised the group here. I'm with Steve and Sarah at a workshop in Arizona talking about astromaterials, and registration with unique identifiers was a big topic. And I've referred to both the RDA interest group and the ESIP interest group, so I think we'll be talking about space a lot more. So I'm excited about the interest that's coming out in that community. So let's keep working on everything, and thanks everybody for the contributions.
Okay,
Yeah, thank you. And also, if your organization is a member of DataCite, we're looking forward to working with you in the community of communities of samples in DataCite.
Okay, well, I'll just do the last. Okay, all right, and we're finishing right on time. Thank you, everyone.
Thanks, Leslie.
Bing bong.
Thanks, everyone.
Bye.
So long. Bye.
Thank you.